show you today that we've actually discovered a clinical candidate going into a phase 2 trial now by screening Rett syndrome. We've screened organoids from the cortical brain region of Rett patients and identified clinical candidates that are advancing now to a phase 2 clinical trial without requiring any additional animal studies based upon the FDA Modernization Act 2. And these are really the first examples of compounds discovered in the human brain organoid that have gone directly into a clinical trial. By developing these human brain organoid platforms, we have the potential to generate large commercial opportunities by making new disease models that are relevant to drug discovery in areas like neuropsychiatry, neurodegeneration, in which there are really no effective models now for screening for new medicines. And then our future plans now are to really utilize these human brain organoid platforms to do discovery of disease-modifying therapies for neurodegenerative disorders, paving the way for breakthrough treatments. I think it's not a surprise to anybody that we're dealing with a human global brain disease crisis now. Brain disorders are the second leading cause of death across the globe, and as many as 10 billion people worldwide live with a brain disorder, and this prevalence is expected to double as the population ages. And in the US alone, the healthcare costs relating to diseases of the brain are around $800 billion. So there's an urgent need for developing new medicines for treating brain disorders. Unfortunately, conventional pharmacological approaches are not really effectively finding solutions, and this is really referred to as the translational valley of death. And so as I mentioned, in 2022, despite $139 billion being spent by the top 20 biotech and pharma companies in R&D drug discovery, there are no clinical trial, no successful disease-modifying therapies for Parkinson's disease. And this is really due to this translational valley of death. This high clinical trial failure rate across all neuroscience indications is due to the lack of reproducible, reliable animal model and cell culture models that can predict human efficacy successfully in a human. And so by being able to develop these new disease models that have the ability to be able to effectively predict human efficacy, we have the potential now for discovering new medicines for brain diseases. So our goal at BrainStorm Therapeutics is really to develop these new human disease organoid models to overcome this translational valley of death. And so basically, CNS drug discovery really needs a paradigm shift. Right now, we're suffering from inadequate models, really fail to predict human efficacy. The other underlying factor is that Parkinson's disease and Alzheimer's disease, they're not diseases, they're syndromes, but multiple different underlying genetic, environmental, and lifestyle factors contributing to the disease pathogenesis. It's unlikely that one drug affecting one target is going to be efficacious across an entire disease population in Alzheimer's disease or Parkinson's, for example. In addition to that, we really have a very limited understanding about individual human validated targets, how they really predict and interact with underlying target biology and the disease processes in which these targets interact, both in terms of intracellular as well as intercellular processes. So we really need to have a new way of studying disease mechanism in a human disease setting. And finally, the conventional pharmacology approaches done by large pharma really is to integrate human biology at the very end of the drug discovery process. And this also contributes to the high degrees of failure. And so at BrainStorm Therapeutics, we're applying patient-derived brain organoids because we believe they're going to be able to transform CNS drug discovery. It provides an opportunity for doing precision medicine. We can take skin cells or blood cells of individuals. We can create induced pluripotent stem cells and then create brain organoids. And these brain organoids are a little three-dimensional tissue that contains all the normal cells involved in the brain. They have native biochemical pathways, and they show spontaneous synaptic network activity. And these organoids also provide a very strong bedrock for doing translational research. We can do phenotypic screening and target-based screening in the organoids and then identify compounds that work in a human disease setting at the very beginning of a drug discovery process, eliminating the need for any animal work. And finally, the recent observation by the FDA, the Modernization Act 2, has now indicated that these human in vitro disease models are sufficient for driving compounds into the clinic based upon efficacy in a human in vitro disease setting. And so at BrainStorm Therapeutics now, we're applying this platform in which we're taking patient-derived brain organoids that are personalized, divided by individuals, iPSCs. They recapitulate complex human disease pathology and provide a personalized medicine approach. But the real strength of our platform is we can use clinically translatable biomarker endpoints to screen compounds and identify efficacy in a human disease model at the very beginning of a drug discovery process. And another aspect of BrainStorm which makes us unique is we're applying artificial intelligence, machine learning technologies to develop advanced network medicine where we can develop disease reactomes, which describe the underlying disease processes that allow us to identify novel targets, biomarkers, and to be able to stratify patients for clinical trials. And we just recently developed a foundation model based upon our Parkinson's organoid work, which has won the NVIDIA Prize for the best platform for doing foundation models. And so again, developing these in vitro human disease models have tremendous commercial potential. You can see that neuroscience indications of even just neuropsychiatry and neurological disorders, it's a $100 billion opportunity for developing new medicines. And within BrainStorm Therapeutics, we have two organoid platforms. We have a cortical organoid platform, which we use for studying neurodevelopmental and neuropsychiatric disorders. And we have a midbrain organoid platform, which contains all the dopamine neurons in the brain relevant to the nigrostriatal pathway, which degenerates in Parkinson's disease, and the mesolimbic pathway that's relevant to neuropsychiatry and addiction. So you can see within our BrainStorm organoid platform, we have opportunities for large commercial potential in discovering medicines for schizophrenia, drug addiction, Alzheimer's disease, Parkinson's disease, and other rare developmental neurological disorders. And so with that as a background, I wanted to show you basically how this platform works. And so we're using the ability to measure transient calcium currents in neurons as a measurement for indirect synaptic network activity. So we can take these brain organoids and grow them in 3 to 4 well plates. Each well has a single organoid and can be identified with individual drugs. And in the middle panel here, it shows the ability to capture the fluorescent signal seen by these calcium transient currents, which generate these flipper waveforms shown here for a healthy organoid. And then we can actually use the ability to dissect all the features of a specific waveform pattern and develop these heat maps, which describe the subparametric nature about how these different flipper waveforms are working. And so with this as a background, we decided to apply this platform to discover new medicines for the rare neurodevelopmental disorders, Rett syndrome and CDKL5 disorders. So I want to describe now the work we did in Rett syndrome to identify clinical candidates that are going into phase 2 trial now. And so Rett syndrome is a rare disorder that affects primarily young girls because it's an X-linked gene. So mutations in the transcription factor MECP2 causes a very profound disease in young girls. They develop normally for the first year or so, but then they undergo a major regression where they develop epileptic seizure activity, they lose purposeful use of their hands, they have problems in the way of the first speaker, right? By mutations in the MECP2 transcription factor. Mutations in this gene cause a very severe disorder in young girls. Over the first year or so, they develop normally, and then they have a massive underlying neurological regression where they lose all the activities which they develop in terms of language, cognition. They develop epileptic seizure activity. They lose purposeful use of their hands. It's a really terrible disorder, and we're using this now to try to identify new therapeutic candidates that can rescue the Rett disease. And so we've been able to generate at scale now in 3 to 4 well plates, we can generate individual Rett organoids, and we can then study them in terms of their gene expression patterns shown here by doing bulk and RNA-seq. We can also then identify the morphological properties of the synapses by doing immunostaining in the organoids. And we've been able to identify now the differentially expressed genes that are seen in cortical organoids from Rett patients compared to healthy patients. And so this is really a high throughput cortical brain organoid platform, which is scalable and can be used now for doing drug discovery. This slide shows actually, we've generated organoids from 2 different rare disorders. Shown in the top are the healthy organoids. The middle panel shows a single point mutation in MECP2, the Q83X mutation, which causes Rett syndrome. And you can see a dramatic difference in the shape of these flipper waveforms between the Rett organoid and the healthy organoid. We also then made a CDKL5 deficiency organoid. CDKL5 is another rare developmental epileptic encephalopathy that causes massive seizures in young girls. And the organoids, again, it's just one single nucleotide change between the Rett organoid and the CDKL5 organoid. But you can see that the organoid pattern is dramatically different. We see a dramatic hyperactivity and a reduction in size of the flipper peaks from CDKL5, and we see this large asymmetric irregular pattern of signaling in the Rett organoids. We then developed a method for doing subparametric analysis of the flipper waveform, and shown in the center panel here is we define all the ways in which the Rett waveform differs from a healthy waveform across multiple patients and across multiple differentiations. And we can then generate these radar plots that are shown here. We can assess all the morphology, the peak heights, the frequency, and the counts. And we actually can determine then a subparametric analysis of how these flipper waveforms differ. And that really is the basis for our drug screen now. We're trying to find medicines that can reverse the unusual flipper waveform back to a healthy waveform. And so again, this just defines how we determine these different subparametric features. And then this slide is meant to show that actually we see a very reproducible genetic expression as well as a functional expression in these organoids. So this slide shows 3 different differentiation batches made over several months, showing a very reproducible, flipper waveform from batch to batch and patient to patient. And shown on the right here is the actual plot of all the differences in terms of the ratio of peak sizes, morphology, counts, etc. And so with that in place, then we were able to determine individual subparametric measurements that is shown in the heat plot. And then we determined a global rescue function, taking into account all the different subparameters to generate a global rescue function. And you can see that different patients and different differentiations show remarkably consistent size of the disease phenotype between the disease and the healthy organoids. So it's a very stable, reproducible platform that shows a robust signal that can be used then for driving drug discovery. And on the right here shows the actual dose response data we see now, where we examine the global rescue as a function of concentration. And so we see a remarkable, reproducible, and robust disease phenotype that can be measured across many different studies to determine a global rescue function. And so we then screen the library from the International Rett Syndrome Foundation called their SMART library. It's a library consisting of about 400 compounds were curated based upon their potential for interacting with known brain pathways and targets that might have some relevance to the Rett phenotype. We screened these individually in single organoids and identified 2 classes of inhibitors that are able to rescue the Rett disease phenotype: acetylcholinesterase inhibitors and HDAC inhibitors. And so we then went on to explore the activity of donepezil, which is the generic version of the marketed medicine called Aricept. Aricept was the first medicine that was approved for cognitive improvement in Alzheimer's disease by Eisai. And so we showed that we wanted to test with donepezil its activity. So previously, Huda Zogby, who first discovered XB2 mutation in Rett syndrome, identified that cholinergic pathways in the brain are very relevant to the disease phenotype. And she tested donepezil and showed it was able to rescue the electrophysiological and the behavioral effects in MECP2 mice. First evidence that this compound might have some relevance in Rett pathophysiology. And so this slide shows that we were able to show that donepezil produces a dose-dependent rescue of the Rett disease phenotype, the synaptic network phenotype shown here in the right. And the ability of the compound to rescue the global rescue function correlates with known concentrations that are achieved in the human brain with 5 and 10 mg doses of Aricept, donepezil. And this ability to rescue the synaptic network dysfunction at relevant brain concentrations also occurs with the relevant pharmacodynamic effect of the inhibition of acetylcholinesterase activity. So we're showing donepezil can rescue the Rett disease phenotype in these human brain organoids within relevant brain concentrations and by inhibiting the enzyme, showing a pharmacodynamic effect. And so based upon these observations, we wanted to also test, while we were doing this work, Acadia developed the first approved medicine for Rett syndrome called trofinetide or DAYBUE. Trofinetide is the 3 amino acid at the amino terminus of IGF-1. IGF-1 was previously shown to have some efficacy in Rett syndrome, at least in animal models, but it has very significant severe side effects. And so Neuren made the 3 amino acids and showed that it had the ability to reproduce the biological activity IGF-1 in Rett animal models. And so in our case, though, it turns out that that trofinetide or DAYBUE, the marketed medicine, does not rescue the Rett disease phenotype in our organoids. And we think we understand why that is. And so here, in the case of donepezil, we have a biological target, a physiological biological target, which we're using to screen to find medicines. Neuren, trofinetide was identified purely based upon symptomatic improvement in patients without any known target. believe now that trofinetide is working to inhibit the inflammatory effects of microglia in Rett patients and Rett organoids. And our organoids are missing microglial cells. And so we've identified now there's a potential for doing combination therapy where we can combine the differentiated mechanism of donepezil together with trofinetide that might have even better improved efficacy for patients. And so this work that we did identifying donepezil as a FLux inhibitor and vorinostat as the HDAC inhibitor, these compounds are both now going into a phase 2 clinical trial that's being funded by the Department of Defense. So we presented this work in the middle of 2022 to the International Rett Syndrome Foundation, and Jeff Newell, who's their lead clinical investigator, wrote a grant to the DoD to test these compounds in Rett patients and achieved the $15 million grant to do these clinical trials. So we're just now filing for the INDs for donepezil and for vorinostat in Rett patients, and the first patients will be dosed next year. When we presented this work at the 2022 neuroscience meeting, several members of the audience, scientists, had daughters that have Rett syndrome, and they've been doing N-of-1 trials with donepezil. And we've been receiving reports, parental reports, that these compounds are showing improvements in language, in concentration, in focus, and sleep, and in even their hand-wringing behavior. So we're cautiously optimistic there may be a benefit to donepezil that's going to be uncovered in the phase 2 clinical trial. Again, I pointed out earlier that both donepezil, shown in the top left-hand corner here, and vorinostat, shown the HDAC inhibitor here, both are able to rescue the Rett disease phenotype in these brain organoids, but they do so through different subcellular mechanisms. And what we did was we identified the subparameters rescued by donepezil and other cholinesterase inhibitors, and the individual subparameters rescued by HDAC inhibitors. And you can see in this slide here that they show completely different underlying recovery signatures at the level of subcellular signaling events. And so this organoid assay can identify differential subcellular signaling pathways that are relevant to global rescue of the Rett disease phenotype at the functional level in the organoids. Oh, yeah, there it is. And so you can see again, across the cholinesterase inhibitors, there's a very high correlation between donepezil and galantamine, two known cholinesterase inhibitors. On the other hand, the cholinesterase inhibitors are very distinct from the HDAC inhibitors, and also distinct from the compound Anavex 273, which actually was shown to fail in clinical trials in Rett patients. And so I'm going to stop there with our work on Rett syndrome, and I'm going to make a little bit of a pitch now for our talk later on today. We've also now been generating midbrain organoids from Parkinson's disease patients. We've been focusing on the two most common mutations that are risk factors of Parkinson's disease, the GBA1 gene, which codes for the lysosomal enzyme glucocerebrosidase, and the LRRK2 gene, which is involved in mitochondrial dysfunction. And so basically, mutations in GBA1 are the most common genetic risk factor that causes Parkinson's disease. GBA1 is shown here, created midbrain organoids from Parkinsonian patients that carry a GBA1 pathogenic mutation. And we've shown that a dramatic loss of dopamine neurons in these organoids by measuring dopamine cell counts, by measuring TH immunoreactivity, and measuring dopamine secretion in the wells. I've been very fortunate to have two really talented people join me as co-founders in BrainStorm Therapeutics, Jun Yin and Maya Castillo here. And over the past month or so, we've been able to generate now the first foundation model based upon organoid data that we've used now to fine-tune the existing pre-trained foundation models that are part of the GeneFormer, SCGPT, and SC foundation models. And by fine-tuning the organoid model data with the foundation models, we've identified a disease reactome now, which is relevant to identifying targets now for Parkinson's disease by doing silico-based computational screening approaches. And we're going to describe that work later on today in our pitch. So with that, I'd like to stop now and introduce our team. So I founded BrainStorm Therapeutics in March of 2023 as the Chief Scientific Officer and now acting Chief Executive Officer. Jun Yin is a very talented data science bioinformatician. Jun and I both work together, not overlapping, but we both work together at Amgen. And Jun is actually a very powerful scientific leader who's been using machine learning and AI approaches all through his career and helped advance compounds into the clinic and onto the market now through his work in the genome analysis unit at Amgen. And Maya has just joined our team as a co-founder from Jean Yo's lab at UCSD. She's earned her Ph.D. in Jean's lab, and she has expertise both in doing generation of human brain organoids, but also in applying bioinformatic approaches, data science, and machine learning to help identify drug discovery. And so again, the take-home messages are that patient-derived brain organoids are a powerful, robust model for doing CNS drug discovery. They're amenable to both phenotypic and target-based screening. Both RET and CDKL5 patient-derived organoids showed unique disease-linked, disease-specific phenotypes that can be quantified across multiple different disease endpoints. And by doing functional screening of the RET organoids, we identified two distinct inhibitors, S-plus inhibitors and HDAC inhibitors are advancing now into a phase 2 clinical trial funded by the DoD. And we've also done, I couldn't show you today, but we've done phenotypic and target-based screening of organoids generated from CDKL5 deficient patients, and we've identified both promising repurposing and novel candidates for the rare genetic disorder CDKL5, which causes, again, a profound epilepsy and autism and mental retardation in young girls. And finally, our midbrain organoids from familial PD patients that have pathogenic mutations in GBA1 gene and also LRRK2 show clinical biomarkers that are amenable to HTS. So our plan now is to use these midbrain organoids from Parkinson's patients to do screening now of targeted libraries that are known to interdict pathways that are relevant both to LRRK2 function of mitochondria and GBA1 function lysosome to identify therapeutic candidates. And we're now going to be using our foundation model to do in silico-based screening to help identify additional targets, to identify biomarkers, and to stratify patients to help improve success in clinical trials. And so with that, I'll pause and take any questions that you might have.
So the ACE inhibitors that's exciting, but have you looked at things downstream of ACE inhibition, like the nicotinic, the muscarinic?
Yeah, we have. And so nicotinic receptor agonists and antagonists have no activity in the model. We were the first to discover that a muscarinic selective agonist is able to rescue the Rett disease phenotype before Karuna and BMS came out with CAR-XT. So we think that's going to be another candidate. We submitted provisional patents now for being able to look at the combination therapies of trofinetide and the CAR-T compound, as well as donepezil and related compounds. We're going to be using our single cell foundation model really to identify the underlying hubs in the disease reactomes that might predict new targets that might identify what the underlying mechanisms are. And so donepezil is a very potent inhibitor of acetylcholinesterase, but it's also a sigma-1 agonist. And so we have tested other sigma-1 agonists that don't work in the model. So we think it's going to be related to the ability of inhibiting acetylcholinesterase activity. But we hope we'll be able to use the ability to dissect the disease reactome to identify the actual underlying subcellular processes, both intracellular and intracellular, that are working through the donepezil. If that helps.
That's great. It's an exciting space right now.
Thank you.
Other questions?
Can you briefly describe some of the machine learning algorithms that have been most successful in what you're doing?
I'm going to defer that question to my colleague, June. So June, would you like to address the question? No, it's okay.
What's working for you?
No, I'm very proud of June's efforts. I really want him to be able to share the light here.
In the recent BioXM hackathon, we have showcased our AI model, which is built upon the published foundation model, but fine-tuned using our organoid data to build a network that can profile the disease model. And we were able to show that our organoid model fully captures what has been changed in the human patient dopamine neurons. By showing they are showing having the dysfunction genes in the similar pathways, and also the foundation model is also able to show that organoid built using different patients with different genotypes that show slightly different signaling pathways. So it's a very sensitive model and showing the disease model really works well for drug screening and can be useful in silico perturbation for the next step to predict the drug candidates and the therapeutic targets.
We're going to give that talk in detail. The last talk today, I'm giving a pitch on that exact work. So if you're here for that, you'll hear the details. Okay.
All right. Well, thank you very much. We'll hear from you again. More detail.